Passaging human iPS cells. Human iPS cells can be co-cultured with an inactivated MEF feeder layer or adapted to a feeder-free substrate with proper medium for feeder-independent culture. This protocol outlines passaging HiPS cells onto a prepared MEF feeder plate. The density and health of the inactivated MEF feeder layer can have a dramatic effect on the HiPS cell cultures. Refer to the table at the end of the protocol for appropriate MEF feeder plating densities for human iPS cell culture. Human iPS cells generally need to be passaged every 3 to 5 days in culture. The appropriate split ratio and plating density will ensure that the culture remains healthy and undifferentiated throughout the 3 to 5 days until the next passage. Human iPS cells proliferate in an undifferentiated state when passaged in small colony aggregates, but not as single cells. It is important to mechanically break the colonies into appropriately sized colony pieces during the passaging process to ensure proper attachment and proliferation. Uneven plating, or plating at high densities, can result in merged colonies after only 2 to 3 days in culture. This can lead to increased amounts of differentiation prior to normal passaging. Likewise, plating exceedingly large colonies can result in poor attachment, merged and/or differentiated colonies. An example of a healthy culture contains evenly spaced medium-sized colonies that are phase-bright, 2D in nature, and able to grow without merging or differentiating until the time of the next passage. Passaging human iPS cells. Step 1. Enzyme incubation. Aspirate the medium from one well of human iPS cells to be passaged. Next, add 1 milliliter of collagenase 4 at a concentration of 1 milligram per milliliter to the well. Incubate the plate for 5 minutes at 37 degrees Celsius and 5% CO2. Step 2. Removing the enzyme. After the 5-minute incubation with collagenase, observe the cells under a microscope and note the edges of the colonies should appear more defined and slightly lifted from the edge of the plate. If the enzyme does not appear to have had an effect on the colony edges, the cells can be incubated again for another 2 to 3 minutes. Aspirate the collagenase enzyme from the well of cells and add 2 milliliters of human iPS cell culture medium to the well. Step 3. Cell collection. To collect the cells, use a 5 milliliter glass pipette. Glide the end of the pipette across the surface of the plate while slowly expelling the medium to gently detach the cells from the plate. This first harvesting step can be repeated 2 to 3 times, but be careful not to over-pipette the detached colony pieces. Transfer the cells to a 15 milliliter conical tube. Human iPS cells proliferate in an undifferentiated state when passaged in small colony aggregates, but not as single cells. It is important to mechanically break the colonies into appropriately sized colony pieces during the passaging process to ensure proper attachment and proliferation. Using a new 5 milliliter glass pipette, add an additional 2 milliliters of HiPS cell culture medium to the well and repeat the scraping technique to harvest the remainder of the cells from the surface of the plate. Transfer the cell solution to the same 15 milliliter conical tube. Centrifuge the cells for 5 minutes at 200 times G. Aspirate the supernatant without disturbing the cell pellet. For a 1 to 3 split ratio, gently resuspend the cell pellet in 6 milliliters human iPS cell culture medium. Mix the cell solution gently in order to create a uniform suspension. Be sure not to over-pipette the cells. The cell pieces should be visible in suspension, but not too large that they settle rapidly. Aspirate MEF medium from 3 wells of the 6-well plate preceded with MEF feeders. Add 1 milliliter PBS to each of the new wells to wash. Aspirate the PBS wash. Step 4. Cell plating. Gently pipette the cells in the tube 2 to 3 times to create a uniform cell suspension. Take up all 6 milliliters of the cell suspension and slowly add 2 milliliters of the cell suspension to each of the 3 wells of the prepared 6-well plate. Incubate the cells overnight at 37 degrees Celsius and 5% CO2 to allow the cells to attach. Remember to monitor your human iPS cell colonies daily to assess overall morphology, growth rate, and to determine the optimal time to passage. To maintain healthy pluripotent cultures, do not allow the cultures to overgrow before passaging. An optimal culture density will allow for healthy colony growth and expansion over the course of 3 to 5 days, essentially preventing colonies from merging before passaging. The irradiated MEF feeder layer can greatly impact the health of undifferentiated HIPS cell cultures. It is important to optimize the plating density for each lot of MEFs used in HIPS cell culture. For a library of StemGent video protocols or to download a PDF of this protocol, please visit stemgent.com.